Introduction to microbial proteases. Proteases are abundantly and widely distributed in biological world including plant, animal and microbes. Proteases group of enzyme that performs proteolysis known as hydrolysis of the peptide bonds that link amino acids together in the polypeptide chain forming the protein. Proteases constitute more than 70% of industrial enzyme alone and microbial sources, bacterial and fungal, are leading supplier of these enzyme. Proteases possess catalytic activity in broad range of temperature and pH. Protease widely used as detergent, in food, pharmaceutical and leather tanning industries. Mechanism of action of protease. The specificity of protease binding is not defined solely by the nature of the catalytic center. On either side of the catalytic center, there is a series of binding sites that favor particular amino acids. Major fungi producing alkaline proteases. Included in this category are Aspergillus candidus, Aspergillus niger, Aspergillus oryzae and many more as shown in this slide, and also, major bacteria producing alkaline proteases are such as, Streptomyces microflavus, Streptomyces moderatus, Streptomyces rectus, and Pseudomonas aeruginosa.
Now is the overall process of microbial enzyme production, which is protease. For upstream process. The process start with inoculum preparation. Medium and microorganism is put into conical flask. The conical flask is then placed into incubator shaker at 200 rpm for 24 hours at 30 degrees Celsius. Next is the fermentation process. Inside the bioreactor, there is a sterile medium same as the medium used for inoculum. The prepared inoculum is poured into the bioreactor. The fermentation process is monitored through the pH, aeration, agitation, temperature, and foam formation.
Now is the downstream process.
The downstream process start with the filtration process. Filtration is to separate the product from the biomass. Second steps is the centrifugation process. Centrifugation is to separate the supernatant which contain the protease with the pellets. Last steps in the downstream process is the freeze-drying. Freeze-dry function, is to make the protease in the powder form. This will extend the shelf life of the protease.
The application of protease are. In dairy product, protease is used for milk coagulant. Besides, protease also used for gluten development in baking. In yeast hydrolysis, protease is used for flavor compound. Generally, protease mainly used for value added in food ingredients.
Created using Powtoon.